Do you want to, oh, did you want to make an announcement about the result of the survey, or do you want to talk about it now?
Oh, okay, yeah. So I swear I will send out an announcement this evening. I just don't want to send one out before tonight's review because I feel like it would have like made a lot of confusion.
Yeah.
So starting next week, per the results of the poll, we are going to start doing our weekly reviews on Thursdays from 12 p.m. to 2 p.m. because that was the time that majority of people voted on. And so I'm sorry if that doesn't work with your schedule. Unfortunately, it's such a big class that like it's impossible to get a time that works for everyone. And that's just the time that most people voted for. So it's the way the cookie crumbles, guys. I don't know what else to say.
That doesn't have to be the end all. If we find that like no one's there, then we can switch back to Friday. We'll talk about it. We'll decide later. But as for now, like Sam said, the next ones will be on Thursday. But anyway, hello everyone. Assalamu alaikum. Let's get started with the first question. I think it's a Gerber question. Yeah, okay. So, all right. Number one, please map the, oh, we have a question in chat. Can we have the link? Yes. Let's see. Actually, maybe someone else could do that.
Yeah,
Please.
I can share the link.
Thank you. So, number one, please map the genes according to the following 3-point cross. Also, let me zoom in. So, just by a show of hands or by a message in the chat, how many of you have watched the video that I put up? Nar, not it. Nar. If you don't watch the video, you're not going to know what's going on, you know, so please go and watch the video. You find it right here in, so LE resources, recordings and documents for exam 3, and it's this video. It's going to make your 3-point crosses like an afterthought. So easy. And I will give you an example right now, actually, but it's not like anything we've done in class before. So I was really hoping that some of you would have seen my method so that we can very easily go through it. So actually, before we start this, how interactive do we want this to be? Do we want everyone to have their own go at it? Do you want me to just go at it and you watch? Or do we want to do it back and forth? Okay. We'll watch for now. Well, Ori, I hope you're eating some good bread. So yeah, if no one has any objections, I can just like bust through it, I guess. It'll just be another example. Please watch the video. Okay, so when I ask you, please map the genes, what am I asking? It's not entirely covered up, but middle gene, that is a component of it. But when you see a table like this that gives you the number of progeny and the genotype, actually, I think this should be phenotype, question mark. Technically, this is a phenotype. Oh, but now my table doesn't look nice and pretty. Now it does, okay. And like the distance between the middle, exactly, there we go. When we're talking about, or when you see a question like this, it is asking you to find the order of the genes and the relative distances between them. Nope, you had it perfectly right. So the first, what's something that we realize about this very first? Actually, let me just ask, what is the first step that we do when we're solving a 3-point cross? And if you watch the video, you will know every step and it will be very delightful and delicious. what's the first thing that we do? Identify the non-recombinants. Exactly. Very good. So who are going to be the non-recombinant progeny? How can you tell who's non-recombinant? Yep, the most numerous individuals. Let me acquire a pen. The most numerous individuals are going to be the one that show non-recombinant or a parental genotype. These are our parentals. And so I guess I can recount it now, but actually, Joseph, like it's, how do I say it? It is technically a phenotype because yes, Ori, there's another component to it that I'm, yeah, phenotype is the actual characteristic that is informed by the information which you find in an organism's genotype. And yes, this is like a collection of alleles. Honestly, I'm just, it's not too important, I guess. Technically, it's a phenotype, but if it makes you more comfortable, let's call it a genotype. Yeah, I'm sorry, I don't really have a good answer for it, but I know from my own experience that it's technically a phenotype. It doesn't really matter. So like Joseph said, the first step that we do is to identify the non-recombinant individuals. I call them parental because, we have our parents. And actually, let's just write them out. So we know our parental individuals because they are the most numerous. And they're the most numerous because it is the most likely that the original parents don't exhibit crossing over. And so we can rewrite these. So we had our original parents that were A plus S M crossed with A S plus M plus. So these are our parental individuals. What do we notice about these individuals that's different from the other 3-point crosses we've done so far? I'll give you a high five if you can identify what that is. Capital letters. No, heterozygous. You're not right, Ori, but you are looking at the right thing. These original parents Oh, Sam, you gave it away. Exactly. Good job, Joseph. These parents are not in cis. They are in trans. So usually when we see parents, they're like, A star, S star, M star, and then A, S, M. Like all of their alleles are of a certain kind, not having a star and then having a star. But these that should be the eraser. These parents are not in cis, they're in trans. They don't have the same kinds of alleles. Yep. So we have identified our parents. And now what is the next step? The next step is to, yeah, I guess I'll wait for you guys. Yep, and then the double crossovers. The individuals who exhibited double crossovers or a double crossing over event are the least numerous. So we have our double crossover individuals. And I guess you can think of it like, just like it is most likely that when you stand in a field, you don't get struck by lightning. That is the most likely scenario. It is less likely that you do get struck by lightning, and then even less likely that you get struck by lightning twice. And in a similar rationale, it is the most likely that you don't exhibit crossing over, less likely that you exhibit crossing over at one locus, and even less likely that you exhibit crossing over at two loci. I had a funnier example in the video. But okay, so we found our double crossovers and we found our parental individuals. So what do we do with this? Well, instead of waiting for an answer, what we do is we compare one of the double crossovers, or I guess someone had an answer. Yep, find which allele is in the middle. Exactly, Casey. That's kind of what we're going for, because when we are looking for our, when we are creating our map, what we're doing is trying to find the order of our genes and the relative distances between them. And so in order to find the order, we have to find which one's in the middle. And the way we do that is by comparing one of the double crossover individuals. It can be this one or this one, but we compare them to the parentals. So let's look at this individual first. So we have A plus S plus M plus. And what we can do is look at this individual and compare it to the parental genotype that is, or phenotype, whichever, that is the most similar to it. And in this case, we have an S plus M plus and an S plus M plus here, but it's the A that's different. And therefore A is the middle gene. Do you want me to give the reason behind that? I could draw it out really fast. It's in the video, very well explained. Okay, we've got a year. So what's really happening when we have crossing over between this parent and this parent? So we have an A plus S and M crossed with an A, S plus, M plus. And so what's happening is that we have crossing over at a locus between the A gene and S gene, and at a locus between the S gene and M gene. And so we have crossing over right here at this locus. So this A will jump down here. Similarly, this A will jump up here. But we also have this M, crossing over at this locus. So this M will jump down here, this M up here. So it's kind of like these two are traveling together. They're travel buddies, and they're going to this side. And similarly, these two are travel buddies going up to this side. So when we look at it, it looks like, when we look at a parent versus a double crossover, it looks like it's the S gene that's different, but in reality, it's these two that are paired that are jumping over. And so that's what we really see when we compare the double crossover having a single different gene than the parent. And that's how we know that's the one in the middle. Watch the video, it'll make even more sense. So I have identified that A is the middle gene. And, oops, that should be the eraser. And so we can rewrite our genotypes. I prefer rewriting genotypes. It makes it more easy to follow. And so we know that A is in the middle. So we have one of the parents is S, A plus, M, and they will be crossed with S plus, A, M plus. It went from trans to cis on the double crossover recombinants. Yes, well, technically, you are right, because, well, trans versus cis only refers to the parents. It doesn't refer to either of the crossing over events or the double crossovers. But yes, actually, no, technically you are right. Like this genotype, oh, not that one, where'd it go? Oh, it's blocked by the chat. This genotype is technically in cis because it is the same kind of alleles. There's just the A with no plus, S with no plus, M with no plus. But usually when we talk about cis versus trans, it is specifically referring to the parents. So Now that we've identified the middle gene, we know that our map looks like this. So now we have to find the distances relative between the genes. And so how do we do that? Just generally. Recombination frequency, very good, thank you. So in order to find the relative distance between, for example, A and M, we have to find the recombination frequency between A and M. And that's been the reason why it has been articulated in class many times. So I'm not gonna go over it here. So we can find the recombination frequency, recombination that happened. Actually, let's not start on the right and go to the left. But let's instead start on the left and go to the right. So crossing over between the S locus and the A locus. It's going to happen like this. I'm going to draw that better. Ah, dang it. Sorry. Maybe it might have been on purpose. So we have S. Oh my God, can I draw this? Jesus, sorry. Ding dong. That was terrible, but it doesn't matter anymore. So we had crossing over at this locus. So, this S came down here, this S came up here. So this is our single crossover event 1. So we can calculate the recombination frequency, or actually we can even redraw the phenotypes. It'd help. So we can just follow our cross. So S went with A and M. Wait, no, that's not right. Oh, because I forgot the plus. Dun, dun, dun. Sorry. I don't always pay attention to what I do. So we had S with A, M plus. It was one of the possible genotypes. We also had, from this crossing over event, S plus, A plus M. And those correspond to genotypes that we can see in our table over here. So S, A, M plus, where do we see that? S, A, M plus, right here. So we have ASM plus, ASM plus, so that's 65. And then S plus A plus M, S plus A plus M, also 65. And so we can do our recombination frequency, 65 plus 65. Thank you, Ari. I think you already left, but thanks for dropping by. Am I missing something? Hello? Instead of waiting, oh, someone answered. Okay, I'll just talk about it. So when we're calculating the, oh my God, never mind. When we're calculating the recombination frequency for whatever recombination happened here at this first locus, we have to consider the individuals that, or all individuals that exhibited crossing over at this locus. So obviously the individuals who only had single crossover 1 exhibited crossing over at the single crossover 1. But in order for these double crossovers to have been double crossover, they crossed over at both loci and therefore had to have also crossed over at this first locus. So we also have to consider them when we are finding the recombination frequency. And we divide this by the total. And I don't have the math pulled up. Oh, you can all see that. Okay, shoot. 0.340. And so that is our single crossover 1. Now we can look at what happened, and now we also Oh my God. So we know the relative distance between the S gene and the A gene. That is 0.340. I'm not sure if Dr. Chiari prefers the numeral or the percentage. Does anyone know the answer? Yes, Casey, you always include the double crossovers when you're talking about what happened at any single locus, because the double crossovers did cross over at that locus. Yeah, she does always say no fractions. So probably go with the decimal when you're answering this. And so we have found the distance between the S and the A gene. Now let's find it between the A and the M gene. So crossing over at this locus, and we can, ooh, no. So we had this and this. So we can, once again, for our single crossover 2, identify the genotypes by following our cross. So we had S plus A M, and we had S A M plus. Those correspond to genotypes we can see over here. Obviously, we've narrowed it down, but let's, for the sake of thoroughness, look. So we have S. I also forgot to write the plus there. So we have S, A plus, M plus is our 32 down here. And then we're looking for S plus AM. S plus AM is 30. So we have our 30 and our 32. And what else? That's right. For double crossovers. Good job. I'm glad you remembered. Divided by the total. That should equal 0.187, I think. And there we go. We have successfully mapped these genes. And we did it in trans, which we'd never done before, but it was nice and easy, right? Right. Wait, what's up? Distance between S and M, we don't need to do. And it's not always going to be accurate, because no crossing over. Well, I shouldn't say that. No, it shouldn't be. Yeah, okay, cool. Any questions? Any other questions? Oh yeah, cis is actually more easy to follow, because in trans we have, our genes that are in trans. The parents are like this, where you have one allele that's, or at least one allele that's not the same as the other, because these two are just regulars, but this one has a plus. And in cis, what you're going to see is that the parents have genotypes that are like the same across the board, where one parent is all S plus, A plus, M plus, and the other parent is all regular. Yeah. Trans is no less, or trans is no more tricky. You just need to like pay attention, do your crosses. Cool. And that took a long time. So moving on.
All right, cool. So, oh wait, Garrett, I don't think I can share my screen.
Oh, sorry. There you go.
Okay, cool. So now I am going to ask you about one of the experiments. I don't know if Dr. Chiari likes to talk about the experiments so much, but I do think that they are important for understanding what is going on and how we found out about DNA and stuff. And so this question is mainly just like trying to make sure that you guys know what Avery, McLeod, and McCarty did. So basically, they injected mice with all these different strains of bacteria, and they injected mice with 4 different strains. So they injected the mice with smooth virulent bacteria, rough non-virulent bacteria, smooth heat-killed bacteria, and a mix of the non-virulent and the heat-killed. So when each of these different types of bacteria were injected into the mice, can anybody tell me what happened for each case? We'll start with A. So what happened when they injected the mice with smooth virulent bacteria? Yes, the mouse died, unfortunately. This one is pretty obvious because the bacteria was virulent, so it had the ability to it was a virus because it was virulent. And so like the mouse was going to die because it got a virus. The second one, what do we think happened to why smooth? So smooth just means that I'm pretty sure smooth meant it was unencapsulated, right, Garrett? And so
Yes.
if you take microbio, you guys will go over like encapsulated and unencapsulated and what that means in terms of like smooth and rough bacteria. But for right now, just worry about like the virulent and non-virulent. So what do we think happened when we injected the mice with rough non-virulent bacteria? Yes, our mouse lived. Hooray. It it was still alive. And then what happened when we injected it with smooth heat-killed bacteria? So since it was smooth, that means it was virulent. So it was smooth, virulent, heat-killed. Yes, our mouse still lived because while the bacteria was still the smooth variety, which should have been virulent, we treated it with heat, which eliminated its virulence. So it was no longer able to cause disease. And then in our last case, what happened when we injected the mice with a mixture of non-virulent and heat-killed bacteria? Yes, our mouse died. And so, can anybody tell me why the mice in situation D died and why B and C still live? Or why, wait, let me rephrase that. Why did die while C still lived? So yes, Joseph, he said it's called transformation, non-virulent bacteria uptake DNA from heat-killed virulent. That is correct. And then Casey, what do you mean by the heat-killed bacteria in C? Okay, so yes, the heat did kill the bacteria before it got to the mouse, but like Joseph said, due to something called transformation, the virulent bacteria or the heat killed non-virulent bacteria were able to transfer some of their genome to the non-virulent, and that transfer of the genome, transferring part of their genome to the non-virulent, made the non-virulent become virulent because some of the genome that got transferred contained the genes that caused virulence. Does that make sense? I feel like that was a lot of word gymnastics, and I just said virulent like so many times back to back.
Can you explain it just one more time?
Yes. Okay, so what happened in scenario D is that our heat killed bacteria, we did heat kill it. So it was no longer able to cause virulence in our mouse because it was heat killed. However, its genome still contained genes that could cause virulence. Do you follow me up to that point?
Yes.
Okay. And so what happens is when we mix these heat-killed bacteria and our non-virulent bacteria together, something called transformation happens where our heat-killed bacteria are able to transfer some of their genome to our non-virulent bacteria. And so when some of that genome is transferred to the non-virulent, some genes that are able to cause virulence are inserted into the genome of the non-virulent bacteria. And so because those genes are in the non-virulent bacteria, they can now cause virulence. Did that make sense? Or did I lose you again? If I lost you again, I can go over it again.
No, I think I follow. I just typed everything you said, so I just have to read it a couple of times.
Okay, if you need me to explain it again, you can just shoot me an email or we can go over it again at the end.
Okay, thanks.
All right, so we know that something in the heat-killed bacteria had to transform the non-virulent to virulent, but what did Avery, McLeod, and McCarty do in order to find out what was in the heat-killed bacteria that transformed the non-virulent? I kind of gave it away when I was explaining, which I didn't mean to do, but like walk me through the experiment that they conducted. Yes, so Joseph said they treated it with protease, DNAs, and RNAs and saw that it was DNA that caused it. And that is exactly correct. So basically what they did is they added RNAs, protease, and DNAs, like this says, to the virulent bacteria. And DNAs was the only one that eliminated the bacteria's ability to cause virulence. So basically when you add RNAs to a solution or to the bacteria, it will degrade the RNA. So like if you take like biochem, which I'm sorry if you have to, but if you do, you'll find out that anything that ends with A's is a protein that degrades whatever comes in front of it. So like RNAs means that you degrade RNA. Protease means that you degrade proteins, and DNAs means you degrade DNA. Biochem is fun. It's just a lot of information for one semester, but I enjoy it. I enjoy the content at least. And so basically, since we know what RNAs, protease, and DNAs is, we added them to the virulent bacteria, and then we found that the bacteria treated with DNAs, when it was injected into a mouse again, the mouse didn't die, like it still continued to live. And so that told us that because the mouse still lived, the heat-killed bacteria wasn't able to still transfer its genome to the non-virulent because its DNA was destroyed. So hopefully that experiment makes more sense now. I know that like when I was first learning about it, I was like, okay, like what on earth is going on? But does everyone understand what this experiment accomplished and why it was important? Okay, cool. I got a few yeses. And like Garrett said, we can explain again at the end if it's not making sense now. Oh, there are only 3 of us here. I didn't know that. Oh yeah, there are only 3 of us. Well, we can explain again if need be. So going on to our next question. This one is just fill in the blank. So DNA is a blank-handed helix. Not quite, it is double-stranded, but that's not what I was looking for in that blank. But it is double-stranded. That is something very important to remember. Yes, I was looking for right-handed. So it's right-handed helix in a what form? Yes, in B form. What other forms did we have? We had two different forms. Does anybody remember? Yes, one of them was Z. And does anyone want to shout out the third one? Yes, I think it was A. So those are our three different types of DNA, or three different forms of DNA, but most of the time you'll find DNA in the B form. And it has two blank strands. Yes, anti-parallel. And so anti-parallel just means that our DNA runs in opposite directions. So we have, I'll type this so we can see what I'm saying. So anti-parallel equals opposite directions. And so basically this means that one strand runs, oh, I cannot type it. One strand runs 5' to 3', and the other one runs 3' to 5'. Can anybody tell me which end of the DNA is negative? Yes, so the side that the end with the phosphate group, oops, is negative. Does anyone know if this is the 5' end or the 3' end? Yes, it is the 5' end. So that is kind of like a good summary of like the structure of DNA. Obviously, there's like more nitty-gritty details, but that's like a good overview and like something to keep in mind for our DNA strands. Actually, one thing that I do want to add is, so we know that cysteine bonds with, or sorry, not cysteine, cytosine bonds with, can anybody tell me what it bonds with? Yes, guanine. And adenine bonds with, yes, thymine. And so can anybody tell me what kinds of bonds hold cytosine and guanine and adenine and thymine together? Oh my gosh, Casey, you're so far ahead of me. You already knew what I was going to ask before I even asked it. So Casey is right. Cytosine and guanine have 3 bonds between each other, and adenine and thymine have 2 bonds between each other, and they are held together by hydrogen bonds. And so these hydrogen bonds on their own are not very strong if you have just like one hydrogen bond, but because we have so many that are connecting our 2 strands of DNA together, it makes those hydrogen bonds super, super strong. All right. So is everyone cool with that? We feel comfortable? Okay, cool. This next question is, oh wow, there are only two of us. All right. So there's the central dogma, which is like very straightforward and very basic, but like something that I think is important to know. So Joseph or Casey, do you want to state what the central dogma is? At this, yes. DNA to RNA to protein. Super straightforward, super easy. So when we go from, so DNA to RNA is transcription. And then RNA to protein is translation. And that's what we're going to be talking about a lot in the next few classes, in the coming classes. So we will have that to look forward to. All right, next question. If a piece of chromatin has 800 nucleosomes, how many histones would it have? So this is something that I don't think we spent like a ton of time on in class, but it's just like something good to know, I think. Yes, there are 9 histones per nucleosome because basically, yes, so you just multiply. So since we have 9 histones per nucleosome, that means we can just multiply 800 times 9, which gives us 7,200 histones. Because, so we have 9 histones because you have 8 in the center. That's our octamer that the chromatin wraps around. And then you have a 9th one that hangs out on the outside that kind of like locks the DNA into place on your octamer. So that one is like it's just like very simple math. Thankfully, the stuff that we're learning now, there's not going to be much math anymore. It's more like conceptual stuff. So if you guys don't like math, you're probably very glad to hear about that. All right. So if we feel good about those, Garrett, do you want to take the next question?
Let's see. Sorry. Kind of as an extension of question 4, we are going to talk about replication. So the central dogma is that we have DNA, which gets transcribed into RNA, which is then translated into our amino acid protein sequences. However, DNA doesn't just go straight, DNA doesn't just go straight to RNA. It also is used to synthesize more DNA, and this is our replication, which is what we're talking about right now. So within replication, there's a bunch of enzymes that are important. So let's please match the enzyme that is used during replication to its function. Shout out any ideas you have. Come on, don't be shy. Helicase unzips, gyrase holds. Let's do one at a time. Okay, so DNA helicase does indeed unzip our DNA. So the way that you can make it so you don't have to just memorize is that we can talk about like the words within the words. So helicase, split it into 2. So we have hele, it has to deal with the DNA helix. It separates the DNA helix. And then we said gyrase holds. And that is correct. Gyrase holds DNA in place while the helicase is unzipping it. And so we have gyrase. When I think gyre, I think like gyroscope, which is like a stabilization matrix. So gyrase stabilizes DNA. Yes, it does. But we'll talk more about that. Or have we already talked about it? We're not going to elaborate on it anymore. So yeah, never mind. I don't want to talk about it. Polymerase adds bases. This is also correct. Polymerase adds the complementary base pairs. Polymerase is what's actually doing the replicating. So we can think back to our cellular biology 213, and a polymer is a repeating unit of monomers. Remember, and so polymerase is going to look at our nucleotide monomers and then polymerize it. So, add on to it. And then that leaves DNA primase. Which obviously makes the primers that begin our replication, because we have our strand of DNA and there's all of our nucleotides on it. It has been, I can't remember if anyone answered your question in class. Oh, sorry. So we have our DNA. Here's the replication bubble. Or I guess this whole thing's the replication bubble. And what we have is little pieces of RNA that go in and say, okay, they tell DNA polymerase to, okay, go in here, start attaching new base pairs that correspond to the base pairs that are already on the DNA. And just look at the base of the word and you'll be able to tell which is what. So now that we know what each of these enzymes do, why do we have telomeres? What is the function of telomeres, excuse me? Telomeres prevent the ends of chromosomes from degrading. Yes, so what happens is that we have, our nice chromosomes. And at the end, we have, choose a different color, we have telomeres. And telomeres are series of repeated DNA that don't code for anything. And what happens is that every time you undergo DNA replication, you have all of these DNA polymerases that are going around. I'm going to need to change that, aren't I? Yes, that are going around and making complementary base pairs for all of your normal base pairs. So you have millions, billions of base pairs, millions of polymerases going around. It's very likely that they aren't going to catch every single base. So what ends up happening is that with every replication that you do, and you replicate DNA whenever you're undergoing mitosis, undergoing meiosis. So whenever you do this, your polymerases don't catch every single one of the billions of base pairs that you have. So, in a replication, you get a little bit scraped off, a little bit scraped off, a little bit scraped off, a little bit scraped off. And that's why telomeres are here, because you don't want replication to be going and digging into your DNA that actually codes for proteins. You'd much rather have it going into these repetitive sequences. And that's why we have telomeres. But eventually, the telomeres, when you've lived long enough and you've undergone some very large number of DNA replication events, you will have eaten through all of your telomeres. And that's when you start eating into the coding genes, and then you no longer properly code for pigment to be deposited in your hair. So then your hair turns white and your skin isn't as elastic and your muscles aren't as strong. That's aging. So actually, Casey, that is an excellent question. A lot of organisms invest heavily into the production of telomerase, which is an enzyme that upkeeps your telomeres. Humans, well, obviously not all cells do it the same way. Certain cells, even within a human's body, have a lot more telomerase than other kinds of cells, and cancer cells highly upregulate telomerase. So cancer cells are kind of immortal in that sense because they'll never experience the detrimental effects of eating all the way through your telomeres and getting to coding DNA because they don't follow the normal instructions and they want to keep themselves going, cancer cells, and so they make a lot of telomerase so that their telomeres last forever. And there are a lot of organisms that invest highly throughout all of their body in telomerase, like turtles, a lot of deep sea organisms. I mean, if you want to, yeah, actually go to every organism you see and just take all their telomerase. Take it for yourself. Can't have mine though.
All right. Do we have any other? Oh wait, Casey asked, oh, can I have some telomerase? Garrett, are you good there?
Yes, thank you.
Okay, cool. So this next question is very straightforward, but it's also just like always good to have practice. So can either of you type, or you can unmute yourself and speak because there's only 4 of us in this Zoom meeting. The sequence of the complementary DNA strand that is typed here. And also include the 5' and 3' in your answer. Yes, Joseph, you are correct. So we know that A binds with T and G binds with C. So we'll have TCAGCAAG for each of the corresponding letters that we have here. And we also want to make sure that we are saying that this is the 3' end because 3' and 5' go together and 5' and 3' go together, because remember that we said that DNA is anti-parallel. Okay, the next question is pretty similar, except we're being asked to write the complementary RNA strand for a different sequence. And so the one thing to really remember here is that instead of thymine, we have uracil. So U replaces T. Yes, Joseph, you have got it. So since we're just replacing U with T, we'll have A pairs with U and G will still pair with C. So we'll have U, C, C, G, A, U, G, C, A. And again, we include our 5' and our, or sorry, 3' and our 5'. Casey, do you feel okay about this too?
I just did it, so I'm checking my answer with yours real quick.
Okay, cool. let me know when you're good.
I got it. I'm good. Thanks.
Okay, cool. All right. You want to take the next one, Garrett?
Yes, thank you. So what is this business of threes and fives? What's numbers? In my biology, I don't think so. Let's talk about it. I kind of just wanted to draw it. So 3' and 5' refer to our central sugar, ribose, it's called. It has to do with the carbons, exactly. So we have our ribose sugar. This is the basis of all DNA. On this first little, and by the way, when you see things like this in chemistry, like all funky shapes, it's just a bunch of carbons that are connected together, and chemists are too lazy to draw all the lines and stuff, how it actually connects. So that's what you're really looking at when you see this. And so we have here at this first vertex an oxygen. Then we have our first carbon. Let me actually write carbon 1. Then we have carbon 2, carbon 3, carbon 4, and then we have another carbon out here, carbon 5. And this is what 3 and 5 is talking about. This is 3', the 3' direction, because it has to do with our third carbon. This is the 5' direction because it has to do with our fifth carbon. And now, also hearkening back to Bio 213, why is this called deoxyribonucleic acid? That's what DNA stands for, is deoxyribonucleic acid. Exactly. I like that diction. So oxygen, or excuse me, DNA instead of RNA. RNA has this hydroxyl on the end, and that's why RNA is just simply ribonucleic acid. But deoxyribonucleic acid has been deoxied. It has had its oxygen removed. And so attached to this 3' carbon is a phosphate group. Attached to this 5' carbon is a phosphate group. These phosphates are what we call the backbone of DNA, because if you zoom out, like if you're looking at a double helix, what you're actually looking at is all the phosphate, this is the phosphate backbone. And then we have our, please switch colors, our ribose sugars. And then even further, if you zoom in, you have all of the, no, I said switch colors, please. Then we have the little nucleotide base pairs. And I also forgot to even draw those. Attached to this first carbon, we have our, please switch colors. We have our nucleotide base pair. Looking at this specific one that I just drew with this specific geometry, which base pairs could this be? It is a pyrimidine. Good job. This kind with just a single hexagonal ring is a pyrimidine. And that is in opposition to a, can you see these? A purine, which has a secondary hexagon attached. And so purines, there's a, oh my God, I can't talk, mnemonic device that states pure as gold, because purines include adenine and guanine. I don't like that, but hopefully it can help. So we have the pyrimidine, and I think of it as like, actually it's a bit more contrived, like pyrimidine, you'd expect it because it has a longer name to like be more complicated. like to have the second one. But no, pyrimidine, despite the complicated name, is just a simple single ring. And these include thiamine and cytosine. And then our purines, like we said, include adenine and guanine. So this is what we're really looking at when we're thinking of DNA. And if we go back to our ribonucleic acid, what about this situation is different? Joseph articulated it before. Yes, and Casey does now. Uracil replaces thiamine. And the difference between uracil and thiamine is that I think there's also a methyl group, and then it's been taken off. And that's the only difference between uracil and thiamine. But even that single methyl group obviously changes what happens because a uracil base pair or a uracil base will provide different amino acids than a thiamine base. So it is important, even though it's very minimal. And that is what the business, a long way of saying what the business of the 5' and 3' end is. Oh, the next question is also me. Okay, let me go back to sharing my screen. And just as another review for Bio 213, what is another difference between DNA and RNA? Just shout them out. If you know a difference, shout it out. RNA is single-stranded. Very good, Joseph, exactly. And yes, Casey, the OH group is present in RNA, but not in DNA. RNA is short-lived. That is also very true because DNA is often single-stranded and because it features, or therefore features, very strong hydrogen bonding between its base pairs. It holds together a lot better and therefore it lasts a lot longer. It doesn't get as easily degraded. And RNA is short-lived because we only need it to, be translated into our amino acid protein sequences. But DNA is the long-term instructions that we use to make our RNA. Okay, so that's the 5' and 3' business. So let me ask you this question. If an organism's DNA is comprised of 13% guanine, how much of it is adenine? Did we talk about acetylation in lecture? And how that relates to chromatin? Okay. 100 minus 13 equals C. Not exactly. I see what you're going for, Casey, but Joseph has it. 13 plus 13 is 26. 100 minus 26 is 74. Yes. Yes. Finish it. There we go. Good job, Joseph. You're a superstar. So what we have is some very basic relationships. So we know that A, adenine, and T, thiamine are complementary bases. They always match together. And then guanine and cytosine, like Sam said earlier, are also complementary bases. And so if we know that an organism's DNA is 13% guanine, there's a better way to write this. So if we have guanine, cytosine, adenine, thymine, if we have 13% guanine, we're going to have 13% cytosine because, they always match up. They're the ones that bond together in our actual DNA helix. And obviously an organism's DNA, like 100% of their DNA is 100%. Like that's where all the DNA is. And it's only comprised of these 4 bases. So if we have 13 plus 13, which is 26 total GC content, that means that the remaining percentage is comprised of A and T. So 100% minus 26 is 74%. And because just like G always equals C, A will always be complementary. It will complement T. They will always exist in equal concentration. And so this remaining 74% will be split evenly between A and T. And if you divide 74 by 2, you get 37%, 37%. And that's what we're looking for. But who discovered the relationships between the 4 base pairs? Mr. Shargoff. That would be Dr. Shargoff to you. Yes, Shargoff. Yes. So let me unveil the answer. Yes. So Shargoff was an Austrian chemist, actually, he became fascinated with the experiment of Avery McLeod. Oh my God. When I was first in genetics, and we were going over the DNA experiments, I thought that my professor said that they were Avery McLeod and the Party, like a 2000s boy band or something. So I always misspeak. So Shargoff was inspired by Avery McLeod and McCarty, not Avery McLeod and the Party, as well as the work of Hershey and Chase, who I think had the coolest DNA experiment. So he was inspired by them, and he's like, okay, so we know that DNA is the genetic material, but what is DNA? Like, what? I don't understand. My brain hurts. What he did was go around and collect the DNA of various organisms, and he isolated the individual, or I guess the holistic amounts of adenine, thymine, guanine, and cytosine, individually. And then he compared them, like did ratios, the ratios that they existed within an organism's genome. And he found that almost always, I think there's even a slide about this in the, please, oh, I'm on the pen. That's why. I think there's also a slide about this. Sorry for taking so long. Dude, scroll, please. Yeah, there we go. So various organisms like E. coli bacteria, yeast, sea urchins, which are, I can't believe it escapes me. Anyway, rats and humans. And as we can see, the ratio between the A and T is almost always 1. It's very near 1 at all times. And similarly with G and C. And that's because A always pairs evenly with T and G always with C. And that was all the questions that we had for you. Did you want us to explain the Avery McLeod and the Parties experiment? One more time.
Since I think I'm the only one who didn't get it, I'm actually going to re-listen to that lecture this weekend. I was going to re-listen, I think, last weekend. Anyhow, I'm going to re-listen. I think once I do that and then I look at the notes, I'll be good.
All right. Yeah, no problem. It wouldn't take any time. We're like 47 minutes ahead of the end of lecture, or whatever this is called, review session.
I have questions on other things as well. I don't know if Joseph has questions about other things too. But if we're in that portion where we can ask questions, then
This is your guys'
have
review
questions.
session.
Okay, cool. So for chapter 8, I just re-listened to this lecture, and if we can talk about some examples, trying to find the slide where it talks about this. Yeah, slide 45 isn't super clear, this concept check. And then also, if we can talk about examples of auto polyploidy and allo polyploidy.
Absolutely. Let me pull up the slides really fast. Okay, here we are. So autopolyploidy is something that we see a lot. Well, maybe not a lot, but we definitely see it in what is the famous example that we've talked about so much this semester. It starts with a K, and there's a dragon somewhere in there.
Oh, the parthenogenesis of the Komodo dragons.
Yes, exactly. That is an example of autopolyploidy. And let me get some free space here. So we have our thing guy. And so we have our Komodo dragon. And this is just the female. Let's say they went through meiosis and they created the, this is their gamete. It's currently haploid. And they're waiting for a male gamete to come up and provide just the right information to create a nice, healthy diploid zygote. But oops, no male shows up because we're on an isolated island and something happened. So what the female does, as we've seen from the case study, is that the female undergoes auto polyploidy. And so the female will replicate DNA and change her ploidy. So autopolyploidy, if we break up the word, auto means within oneself. Poly means many. And ploidy is all about the folds. The fold, oh my God, I did it again. Pretend that says folds. And so the folds is all about, the like funky looking chromosomes, the way that I draw it. So when you think of ploidy, think about like bodies of genetic information. Many, so having not just one chromatid, but maybe two, maybe even 3, 4 chromosomes. Thanks for coming, Joseph. Always make the review so worthwhile. But yeah, so we have polyploidy, which is having many bodies of genetic information, many chromosomes, and auto happening within oneself. So just by herself, this female Komodo dragon changed her ploidy. Polyploidy is like a change in ploidy, changing the number of series of genetic informations you have. So this female within herself changed the amount of genetic information she had and therefore underwent autopolyploidy. Was that what you're looking for?
Yeah, so that makes sense. But so can we could we say that pretty much autopolyploidy is when fusion of maybe not fusion, no cell division. I'm looking at the mitosis and meiosis example. So it looks like non-disjunction for meiosis.
Yes,
the cells replicate, they don't split apart during anaphase. So then you've got, or maybe they do split apart. No, it looks like the homologous chromosomes, they just because there's two anaphase. So in anaphase 1, I guess they didn't split apart. And then it looks like anaphase 2, they did. If I'm reading that right. On slide 49.
Slide 49. Let me see. Yes, you're exactly right. And so let's say that we have this plant cell here, and they're currently 2N. So we have this non-disjunction event, and then we have meiosis 1 here, or excuse me, that's not correct. We have meiosis 1 here, and then this will go. Did I do that right? Oh no, I did not do that right. So we have meiosis 1 here,
Mm-hmm.
kind of, and then we end up with this is as our product of meiosis 1, where we should have had, you know, 2 cells. Pretend that's a good drawing, please. And then in meiosis 2, let's say that it goes off without a non-disjunction. So we end up with this when we should have ended up with this. So this cell, this gamete has twice as much genetic information as it should. And so let's say if this organism then fused with a gamete that was the regular N, they come together and form 3N. So like I said, autopolyploidy. Polyploidy is a change in ploidy. And auto, within oneself, within their own meiotic processes, they changed their ploidy from 2N to 3N. And that is another example of autopolyploidy.
Okay.
Yeah, just like you see on slide 49, actually.
Okay. And then can we talk about allo
Yes, of course.
polyploidy?
And it's kind of, oh, that's what happened. The pen went away. Okay, that's better. So kind of as an extension of that, we can have, let's say, I'm just going to redraw it actually. And Sam, feel free to jump in. Let's say we have another plant, because plants are famous for this. We have a plant cell that is 1, 2, 3, 4N. And then they'll undergo meiosis. And then let's say that it goes off well. And then these two split up into this. And then even their meiosis 2 goes well. And they'll split up into haploid cells. Remember, haploid relative to the original number. So we started with 4N, and these gametes are 2N, which is half of the original, which makes these relatively haploid. And let's say that this plant, this gamete, contributes its 2N genetic information to another plant who started out as 1, 2, just 2N actually. Oh wait, dang it, I messed that up. There was a better example, but you know,
why am I thinking that meiosis 2 ends with 4 haploid cells? Oh, because we are starting with 4N, that's why. Never mind. Okay, I'm good.
Okay, so let's say that we have another plant that is 8N, and it also splits up into 4 haploid relatively cells, or haploid gametes. So this 4N gamete is going to contribute, and this 2N gamete is going to contribute. And so we have 4N plus 8N, or no, excuse me, that's not 4N. We have, excuse me, a 2N gamete plus a 4N gamete. They combine together to make an overall 6N organism. So we had an example of allopolyploidy. Remember, polyploidy is a change in ploidy, or a change in the amount of genetic information you have. And allo is a prefix that means different. Kind of like if you remember AP Bio, you have allopatry and sympatry. Sympatry, sim means together or same, and patry means fatherland. Allopatry is therefore a different fatherland, and that's talking about speciation, like where different species come from a different fatherland. So allo, that's how just I remember it, but allo means different, and polyploidy is a change in ploidy. So you have a change in ploidy coming from two different sources. That's what allopolyploidy is. Plants are famous for this because they can handle it. Animals can't really do this.
Okay, so auto is change in ploidy due to self. Allopolyploidy is change in ploidy due to hybridization between like two organisms or mostly plants with two different types of ploidy to create an organism that's a combination of the two.
Exactly.
Okay, makes sense. Alrighty, and I think so that would explain why on slide 51 that plant ends up 2n equals 6. The plant ended up with 6 chromosomes because that's the offspring of basically a hybrid cross.
Mhm.
Yeah, I think last question is in regard to concept check 6.
Let me look for it.
It's slide 45 in my deck.
Do you want to explain this, Sam?
Yes, what, I'm sorry, what chapter was it?
It's chapter 8,
8.
slide 44. Sorry to interrupt.
Chapter 8, slide 44. Let me pull up the slides. All right, this slide.
Yes.
Okay, so briefly explain why sex chromosome aneuploids are more common than autosomal aneuploids in humans and mammals. I think so. Obviously, like the answer is down here, but so basically what happens is we know that dosage compensation is like a thing that happens, right? Like do you understand dosage compensation and like why it occurs and why we do it?
Yes, that's in regards to the X chromosome, right?
Yes. So like if you have like 10 X chromosomes, you're going to inactivate 9 of them and you only have one active X chromosome. And so it's much easier to live with like many X chromosomes because you inactivate all but one of them. And so you're still a very like normal and functioning human being who doesn't really have any problems. So like you could be XXY or you could be triple X or quadruple X. Like all of those individuals will still live a very normal life, right?
Right.
But if you have, for example, like trisomy 21, where you have 3 copies of your 21st chromosome, you end up with Down syndrome, which is very like it, you can't live a normal life with Down syndrome. It causes a lot of issues. And so if you have a third copy, or if you only have one copy of any of your autosomal chromosomes, so those are your non-sex chromosomes, then you can end up with disorders like Down syndrome and many others. And I think a lot of the time, if you have trisomy of most of almost any of your other chromosomes, you don't live, but there are four that I think you can still have a viable birth. I think it's like 8, 13, 15, and 21. That's probably wrong though. But for the most part, only trisomy 21 tends to be viable. And even if you have trisomy 21, like you end up with Down syndrome. Does that make more sense now?
Yeah, that makes sense. I guess I'm just curious why sex chromosome aneuploids are more common than autosomal ones.
So basically it's just more common because you can create more viable embryos that have that are sex chromosome aneuploids than autosomal. So when you have an autosomal aneuploid, most of the time it'll undergo spontaneous abortion and they just won't, like, you'll probably never even know that it existed.
Okay.
Were there any other slides that you want to go over from chapter 8?
Not yet. No, I think that's all for chapter 8. I'm still gonna re-listen to a couple lectures, but that's all for now. Thank you both.
Of course.
Thank you. Dang, we're 30 minutes early.
I know. I don't know what to do with ourselves, as we could go over literally anything if you wanted to.
I kind of want to talk more about the DNA experiments. They're kind of my favorite part of genetics.
Actually, yeah, the DNA experiments are really cool. I remember when I took the class with Dr. Shear, she like absolutely loves the experiments. So she always asked us questions about the experiments on quizzes and exams.
I know,
We
yeah.
can talk about that if you want. I actually looked at real fast. Doesn't translocation seem a lot like crossing over?
So translocation it doesn't have anything to do with crossing over. Translocation happens by an unknown mechanism. Like it's genes that just, like wherever they are, they somehow find the inclination to like get up and locate themselves somewhere else in the genome. And we don't know why or how it happens. And it leads to, like, it's probably the most important thing that happens in our genome, but we just don't know about it. Crossing over, however, is like a very obvious phenomenon that we can see happen.
Okay. But yeah, if you want to go over the experiment again, I'm here to. I'm here. I don't have plans.
So this.
Yeah.
So something that I don't think was, I guess it's fine. So what we had is our guys. And what wasn't said about this is that, when you heat kill a bacterium, like the DNA that was inside of it kind of just like floats out and is available to anything. And another thing that wasn't articulated is that bacteria, they like to share their genetic information with surrounding bacteria. That's why they, in quotations, evolve so quickly, because they're constantly acquiring new influxes of genetic information that help them to survive. And so what happens is that, for example, in this mouse, that was kind of like the key to the whole experiment. What you had was heat killed bacteria. So a bacteria was killed by heat and it busted open and it died. So its DNA just floated out. And then there was another bacterium that intook this DNA. And this bacteria was previously non-virulent. Virulence is just like how infective something is, I guess in this case, how harmful it is to a mouse. So it was previously non-virulent, and this was a virulent bacteria that was died, and its very harmful DNA was then leaked out into this test tube, and then a previously harmful bacteria intook this harmful DNA and became virulent. And it's this virulence via the DNA that causes the mice to die. And so that's how we knew that it was the DNA rather than the RNA that was doing the stuff. Well, I guess maybe what definitely sold the deal was the like DNase, and DNase is an enzyme that destroys DNA. And so if you put that DNase into this vial D, then the let me bring that back, sorry. If you put DNase into this mix, DNase is going to run around and it's going to chop up all this DNA so that it can't be harmful no matter what. And therefore, even though this non-virulent bacteria became virulent, its virulent DNA, like the DNA, the stuff that was making it kill the mouse, was chopped up so it could no longer kill the mouse. But my favorite experiment was definitely Hershey and Chase. Where is it? Right here. So what they did was they had some phages. And a phage is like, do you know what a phage is?
No,
Have you heard of the
I
term?
do not.
So a phage is a kind of virus, and I'm going to draw it very poorly. I remember it from I have a very deep memory of like an episode of Jimmy Neutron where these guys were running around. I don't know.
That is the exact same thing that I think of
Yes.
every single time I think of phages.
Oh, I'm so glad. Oh my God, Sam, you're too cool. But yeah, we have these guys. They're little viruses, and what they do is they have like a little drill kind of thing. Should I redraw this? Nah, it's fine. We have a little drill kind of thing, and they have their DNA. And then when they're ready, they'll inject this DNA. They'll be like standing on top of the bacteria and they'll inject it into the bacteria. And so what these guys did was that they took these phages, and what they did was, excuse me. Oh, actually, I should also articulate. So they had some radioactive isotopes, and we use radioactive isotopes because they literally, they're radioactive. They glow. They emit photons because they're unstable, and so we can always track the progress of an isotopic atom. And so they took an isotope of phosphorus, which you find a lot of in DNA. I'm going to color code this too, by the way. So they took some radioactive phosphorus, and you find a lot of phosphorus in DNA. Remember, phosphorus was like the backbone that connected to our sugar and stuff. There's a lot of phosphorus in DNA, and they had a radioactive isotope of it so they could always track its progress. And they had a radioactive isotope of sulfur, which you find a lot of in proteins. So what they did was they had some bacteria, and they let the bacteria inject their genetic information. They didn't know necessarily whether or not it was proteins or DNA. So they let the bacteriophage inject their DNA into the bacteria, and then they threw it in a blender so that it would like chop the phage off of the bacteria, and you'd end up with just the bacteria at the end with the genetic information from the phage, the virus, injected into it. And then they like centrifuged it. And when you centrifuge stuff, like it's separated by its density. Like if you centrifuged water and oil, because water is more dense than oil, you'd find water at the bottom of the test tube and oil at the top. Similarly, you had, in the case of the sulfur, because, they found that this was, so just like water and oil, bacteria are obviously heavier than a virus, a little phage, because a little phage is thousands, millions of times smaller than even a bacteria. So you'd find bacteria at the bottom of a test tube after you've centrifuged it, of course. And so what they found was that in the case of the sulfur remained at the top because the sulfur, which is a part of a protein, stuck with the viruses, which are less heavy than bacteria. And so you'd find the viruses and therefore the sulfur at the top of the test tube. But with the phosphorus, they found the phosphorus at the bottom with the bacteria because they knew, therefore, that it was DNA that the virus had injected into the bacteria. And that DNA is what viruses use, or what this virus used to rewrite the instructions of the bacteria. And that's how they figured out that genetic information was passed along by DNA rather than protein. And viruses can be made up of double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. But for this example, they had, I think, single-stranded DNA viruses. And then we had our guy Shargoff. Oh, I hope we talk more about Rosalind Franklin. Do you want to talk about her, Sam?
Sure. I mean, I don't have much to say about her other than she did the X-ray diffraction stuff, and Watson and Crick were beefs and stole everything that she did. Yeah, that's pretty much that's what I have to say about that. She found the structure of DNA, basically, that it was a double helix from X-ray diffraction, and then Watson and Crick stole her information, or like her findings and like a bunch of other people's findings, and like put it all together and were recognized for it when they were just thieves, really.
Yeah.
But
So Rosalind Franklin worked in a lab with a man named Maurice Wilkins. And because she was a woman in the actually, I don't even know the date. I think it was the 50s. But yeah, because she was a woman, she knew that her research findings wouldn't be as well respected. So she really wanted to make sure before she published anything. And she was getting some abnormalities in her data. And so she was also very paranoid that her information wasn't correct. But what she was discovering was the A and Z forms of DNA, which have a different shape than the B form that we usually see. And so she was kind of freaked out by that. And that's why she didn't publish the findings herself. But one day when she was out, I don't remember why, she was out of the lab, and what are their names? I forget. I dislike them so much that I have blocked them out of my
Watson and
memory.
Crick.
Thank you, Watson and Crick. Watson and Crick paid a visit to Maurice Wilkins, who worked in the same lab as Rosalind Franklin. And Maurice Wilkins showed her the images, or excuse me, showed Watson and Crick Rosalind Franklin's images of the X-ray crystallography experiments that she conducted. And they're like, oh, let's steal it and not give her credit. Of course, they credited Maurice Wilkins with their findings, but not Rosalind Franklin. And Rosalind Franklin, because she worked with X-rays a lot for a very long time, she used to experiment on coal to look at the structure of carbon rings and stuff. And then she transitioned to DNA, but she'd been working with X-rays her entire scientific career, and not always protecting herself from the radiation. So she was very highly radioactively dosed, and she developed a horrific case of ovarian cancer. And so she died even before Watson and Crick published her own findings, and then they didn't credit her for it. And they won the Nobel Prize. And yeah. Actually, one of them the one who's still alive, has been stripped of all of his awards and honors because he's such a terrible racist. He is a strong believer in eugenics, which is like the concept
Of
that,
breeding humans.
yeah, for exceptional characteristics, and that's often based on false perceptions of race. And so, oh my God, there are documentaries about how horrible this man is. But yeah, that's a long story. Well, I don't see the harm in cutting it 15 minutes early if you don't.
I am also okay with that. Casey, do you have anything else that you wanted to go over?
Good. Thank you guys.
Oh, thank you for always showing up and asking
Absolutely.
questions.
Gotta get my questions answered somewhere, somehow. Otherwise, I'll just sit there and look at the PowerPoint and rewind the lecture 1,000 times.
Oh, just personally for you, does the new Thursday time